a very, very brief background on predicting annotation gene structures. Again, not to insult anybody's intelligence, but rather to bring to the front of the mind of some of you that may not be as familiar with it. I'll discuss over the next few slides a little bit of information about how these genes that we're going to use in Apollo obtained and why is this important. So, you know, the basic premise behind gene prediction annotation is that we're trying to identify genomic elements primarily by focusing on protein coding genes and using the composition of the DNA to do this, as well to identify several other elements, repetitive elements, and other RNAs. And this process happens in two steps that I'll describe briefly. This all comes from information that was nicely put together by Mark and Daniel a couple of years ago, Mark Yandel and Daniel Enns. So in that computational phase, we have experimental data that are aligned to the genome, you know, so RNA-seq reads, for example, are aligned to the genome. As well, there is the possibility of doing these gene predictions, generating them ab initio, just simply based on the nucleotide composition, and or using as well experimental evidence to identify domains and motifs. And what you would end up with is something like that bottom gene drawn from the gene prediction. In terms of ab initio, many of you are familiar with this, but you deal strictly with the genomic sequences and use statistical approaches to be able to find coding regions and also to identify typical gene signals. And some examples of this are the predictors listed below, like FGen Sage, Augustus. And then we can also use experimental evidence in order to find genes. And so for this, we're using sequence similarity searches against public databases. Eventually, what we're going to get out of this computation phase is, you know, the single most likely coding gene. And that's what those gene predictions would look like if we're using things like SNAP or Exonerate in order to make the predictions. Later, then we're going to be able to associate all of these experimental data in order to make a more reliable structural annotation. And the final result from that will actually be the gene models that look more like the ones that you're going to have as the starting point in your Apollo instances, which are gene models that include untranslated regions, isoforms, and several evidence trails, for example, like the alternatively spliced two isoforms that are at the bottom, represented in the light blue color. Then, as Anthony was talking about, you'll get a consensus gene set that can either be made using combiners such as Evidence Modeler from the Broad Institute, or by using packaged tools into pipelines like Maker, which has been used for this particular project. There are several different things that are positive about each of these approaches, and there are other not so positive. And so the suggestion is to basically go in some type of hybrid approach in which you do some reference guided and ab initio gene prediction in order to get those gene sets. So two words on annotating gene functions. This is not the objective of this chat today, so I'm not going to dwell on that very long. But in order to annotate gene functions, we're basically associating metadata to all of those gene structures. And you can do this in Apollo by using our tool that connects directly to the Golar database from the Gene Ontology Consortium. So you'll do these assignments of function to the putative structures. Just be mindful that sequence similarity approaches by themselves without any evolutionary context to those searches are less informative than when you're using a score value. That's why I was asking earlier whether InterProScan was involved in this process of assigning the gene ontology, and it is. So here's a flash refresher of the three main components of the gene ontology and how you'll be associating these terms to your genes of interest from either of the three sections, the molecular function or the localization of a gene or gene product in the cell through cellular component, or in a more broad manner, a commonly organized series of events through biological processes. All right, what you came for today is Apollo. So we're going to learn a little bit more about how to collaboratively curate gene structures. I'll give you a little tour of the Apollo window for the next few minutes, and then I'm going to try an experiment in which I actually introduce you to the Apollo window, kind of a baptism through fire with a webinar. So we'll try this together and see how it works. You'll be my guinea pigs. This is the Apollo window. Apollo is built on top of JBrowse, as Anthony was saying before. So we're basically a plugin sitting on top of JBrowse, and JBrowse is a web browser genome. This allows us to conduct collaborative and instantaneous annotations all on the web without having to download anything. At the top, the big blue arrow on the left denotes user-created annotations in this little yellow area. And down at the bottom, both on the side panel and in the larger area that cannot be edited, we have evidence tracks that depict information such as, in this case, some stage and cell type specific transcription data. So you can see some RNA-seq reads in green and lighter green, and then some gene models, XY plots, and some heat maps. You can also upload your own evidence, and we can discuss more of that later on. It'll happen in a per session type of environment. Say that you have some transcriptomic data that you want to align to the genome, and you have the GFF3 alignments to it, but you're not ready to share this with everybody because it was just a small experiment you were conducting. You can do this on a single session basis, and it'll go away once you log out, but it'll allow you to see some data aligned to your genome. And you can also change the color of the coding sequence by frame. And so I'll show you that in a second, but you'll be able to color all of those coding sequences. You can interrogate the genome using sequence similarity searches. You can help using these little tools and magnifying glasses and arrows to navigate and zoom through the genome, as well as use this box attached to the very top of the JBrowse window to search for a gene model. I'll give you some other ways to do that. On the right of the window, then there's something we call the annotator panel, and I'll give you a tour of those in a second, but to give you then a sense of what you can see, you'll be able to visualize pseudogenes and regulatory elements, as well as protein coding genes, and annotate them in that user-created annotation area given the structure of the experimental data that you have. And there are many, many different functions that Apollo can perform. We have 10 different types of annotations that you can conduct in that user-created annotations area. And then on the right, I have an example of all of the functions that you have from that right-click menu, which we'll go into detail in a little bit. And then once you're done with all of this work, then you'll be able to export all of the information, both as GFF3 files, as well as GFF3 with FASTA, or simply by exporting the sequences of those proteins that you're most interested in. And of course, in collaboration with your admins, you'll be able to get information about the entire genome and exporting some data from the entire genome for those global analysis. The great thing about all of this is that you can perform many of these functions because in your computer, basically the editing logic is working on your client, on your browser, and that's what this little researcher sitting down at the computer depict. But also every edit that you conduct is immediately and automatically saved to a centralized database, which allows you to work with, for example, me over here in California, looking at the same gene model, and we can have a discussion about what the best structure is going to be while we sit apart. And so this is collaboration and is in real time. Later on, if you guys are interested in, I can go into more detail regarding the Apollo architecture. For the majority of us, it's just suffice it to know that we have three components. We have the web browser in the front, so that's the client that you're interacting with. We have an editing engine, which is all of the logic in the brain of Apollo that thinks in biological ways, so that when you conduct a modification to the gene structure, the biology is respected. And then you have a database, versatile allows ways of storing these data and then to make it accessible to you so that you can query it as well. If later on some of you are interested in setting up instances of Apollo for your own laboratory for other genomes, please feel free to contact me and we'll be able to help you through that process. In general, then, when we're thinking about the process of curation, then we're thinking about these 6 components. We find the region of interest that we want to work with, either because we have a candidate region or because we have some sequence that we've used to query the genome. We select all of the appropriate evidence, and then we compare that into that region. We figure out whether or not a genomic element or a feature it is a reasonable start for a gene model. And if necessary, we adjust that gene model. Once we're done and through the entire process, we conduct a check for integrity and accuracy of this representation of the underlying biology, and then we're done. We comment and submit it for the next annotation. Sounds easy, yes? Okay. So I just want to draw a baseline. Again, no intentions of insulting your intelligence, but simply to bringing attention to a few things that we'll need to clarify or at least agree upon when we're conducting annotations using Apollo. So I've taken the liberty of defining 3 very basic things. I know that there's a lot of argument, 150 years worth of discussions on what a gene is. And so for the purpose of annotating a gene in Apollo, we're going to consider a gene as the genomic sequences that directly encodes functional products. And if there are several of those functional products that are sharing overlapping regions, then we're going to take a union of all of those overlapping sequences that are coding for them, and we're going to make sure that this union is coherent. So these products can be separately processed, but it does not require that all products necessarily share a common subsequence. This sounds very gibberishy if I just give you the rules this way, so the best way to interpret this is probably an image. So we define a gene as the union of those genomic sequences encoding a coherent set of potential overlapping functional products. Then at the very top, the DNA as it lies on the chromosome we have delimited all of these 5 elements, A, B, C, D, and E. The primary transcripts that we obtain from those 5 elements can be something like the ones depicted in purple at the top. One primary transcript that includes elements A, B, and C, and another one that includes D and E. Yet a third primary transcript that includes some region on the left and region on the right that are called X and Y that overlap those other primary transcripts. When these transcripts are spliced, for example, in this particular demonstration, you have a few transcripts, one that, for example, has sequences from A and B, and another one that has sequences from B and C, and these are all the coding sequences that eventually become the proteins in red. So you can see that in the left part, all of these 3 proteins are actually sharing some coding sequence. Either they share the A or they share the C. But on the right, the protein products, the functional products that are being translated out of these spliced transcripts actually share an untranslated region but no coding sequence. And so because of this, then we're going to say that elements A, B, and C are all the result of one gene. Elements D and E are 2 different genes. And then that non-coding RNA at the very bottom is gene 4. And that will be what we'll have to take into account when we're conducting these structural annotations. I know that there may be strong opinions about this, and if there are, I'll be happy to have conversations on how to improve this model. This is your best friend through the entire process of curation. Just one single reminder, untranslated regions are part of your exon. So if you have sufficient evidence in order to supporting this untranslated regions, I invite you to go ahead and include them when you're annotating your gene. And just a quick reminder, the open reading frame, there will only be 1 reading frame per section of DNA that is biologically relevant at a time. And with that in mind, we'll keep in mind that Apollo constantly recalculates the open reading frame to try to reach the longest ORF after each one of your edits. So keep that in mind. And then this little nugget of knowledge, when you're using Apollo, you will always have the ability to know whether or not a non-canonical splice site is being used. Sorry about the train in the background. If you're an intern, you'll have a 5' end splice site, a splice signal that usually is a GT in some arthropods, and so you may actually come across this. You'll have a less common GC donor. And then on the 3' end of your life, if you're this intern, you'll have an AG. So this is what you're expecting to find at the beginning and end of the intern. You'll find a GT, then there's the intern, then there's an AG right before the next exon begins. Take a picture, download it, memorize it, write it, tattoo this in your arms. You're going to need this. And this, of course, is what has the ability to give us alternatively spliced isoforms. Lastly, remember that interns can interrupt the reading frame at any given point. It could be by inserting themselves between 2 consecutive codons, or between the first and second nucleotide of a codon, or between the second and third nucleotide of a codon, which means, of course, that each of those exons are not going to all be in the same frame. I know that this is biochemistry from the beginning of the times, but this is stuff that bites me every time when I start looking at a gene model. So remember these because the frame will change from one exon to the next. There are several obstacles to transcription and translation in nature, and some of those will actually have some consequences on how the genome is organized when we're trying to assemble it artificially using our computers. And so some premature stop codons in the message are possible, and this is simply because the machinery is trying to check and correct all of these potential processes, and this actually can cause some changes in the reading frame. So beware of those frame shifts as well. Indels can give you headaches, and so make sure that the peptide is not abnormally long or short when you're looking into these sequences. Let's go and explore now this window a little bit more. This is now the part in which we talk even more in detail about the functionality of Apollo. And then after a few slides, I'm going to invite you to follow along and play with me on our testing servers, training server. So, this is the basic data visualization. At the top, that yellowish part is the user-created annotations area. At the bottom, all of this information are evidence tracks. This could be RNA-seq reads or any other reads aligned to the genome. This could be gene models from other organisms that have been aligned to your genome. This could be several other tracks of, you could have some VCF tracks, you can have some SNP data, anything that can be aligned to the genome and put in a GFF3. Okay, I'm going to qualify that anything. Most things that can be put into a GFF3 file can be visualized. And then on the right, you have the annotator panel. So let's explore that annotator panel on the right a little more. If you have administrative privileges, then you'll have this organism, user groups, and admin tabs. Most of you are not going to be able to see this, so I'm just going to concentrate on the tabs that describe information about the annotations, information about the tracks, and information about the reference sequences, which in this case are the fragments of the assembly. So we're going to zoom in to this upper right part of the window. So this is what the annotations tabs looks like. It allows you to navigate the genome in a more versatile way than just trying to find the gene and trying to find the group. Annotations that are already existent are going to be listed. See, I don't know if you can see my arrow, but in the bottom left of this window, we have a little blue arrow that indicates where the gene is located, so the parent feature in this case, and then a little green arrow indicated where the mRNA is visualized. Okay, so here we have, if you click on that green mRNA, over in the upper right part, you have the details, and so that's what that number 1 is demonstrating. You have information about the particular gene model, and then in the bottom right, on the number 2, you have some details about the exon boundaries. You're able to either correct the structures from these windows, or you can do it directly from the gene structure. You can also use this tab to navigate to an annotation. This is the same window, simply I'm highlighting 2 different things. Look at this big blue button that says go to an annotation. If you click on one of the annotations on the genes, or if you have actually double click on that gene to reveal the mRNA, you'll be able to click on either the blue button at the bottom or the blue button on top saying go to an annotation. You can also search for an annotation using the boxes on the right. On the far right, I've highlighted a little detail where I show you that the drop-down menus will give you the ability to look for only pseudogenes or only transposable elements or any type of annotations that you are conducting. Then you can also display all of the different tracks of evidence data. So if we move to the next tab, the tab called tracks, we have on the left part, we have two arrows. There's a purple arrow showing us going right, and that one, as well as the upper right panels, allows you to show and hide JBrowse track selectors. What are these? So the examples that Anthony was showing you actually reveal the tracks as the ones that come native with JBrowse. The arrow points at this list of available tracks, and that's what the JBrowse environment looks like. We have enabled the ability for you to look at these tracks in that way because there are some nifty features that come with JBrowse. You likely won't have to worry about this. Anthony and Fabrice and all of your team have gone through a lot of trouble to set up a really cool instance of Apollo. So you don't have to go and look for the selectors, but I just wanted to explain what those were. Under the tracks tab, the pinkish burgundy arrow in the middle is showing a checked box from the list of tracks. So checking and unchecking will allow you to reveal or hide a track. You can also do that directly from the annotation window by just checking it off, just marking it off. And you can also navigate to a specific reference sequence. So in the context of Apollo, all of the fragments of the assembly are called reference sequences. These could be scaffolds, these could be chromosomes, or in the case of this example from the honeybee genome, where scaffolds have actually been anchored to chromosomes, we call them linkage groups. So they've been anchored to linkage groups. There are 16 of them in honeybee, so these are called group 1 through 16. And because they're not completely assembled, rather, yet they're fragmented, then there's like linkage group 16, 1 through 33, for example. And so 16.1, 16.33. So that's what those arrows are, marking the different ways in which you can navigate to a sequence. From the annotator panel on the left, you can do this from the top by typing in the scaffold you're looking for, by searching it with the arrow in the middle, or at the very bottom by double clicking on the name of the group or the reference sequence that you're looking for. You'll navigate through that. If you go to the annotation window, to the actual place where you're conducting the edits, you can see these two inlets on the right showing you that you can navigate to a different reference sequence by simply using the drop-down menus or by typing its name in the search box. This will become easier to understand once you're familiarized yourself with the window. There are several other things you can do. You can share the location that you're looking into. You can also switch organisms. If you have permissions to work in more than one of the 4 species or all 4, then you'll see a drop-down menu. See on the middle left part, the arrow pointing to the word honeybee. If you see that purple arrow coming out of it, you see that there are 3 organisms listed. I have an example in which I can see the human genome, the cat genome, and the honeybee genome. You can also hide and show the annotator panel. On the far bottom right, you can see a little caret, sideways more than signs, that allows you to just hide or show the annotator panel. And then on the bottom left, we have the ability to end the session that we're working with by either looking, clicking on your email, hitting log out, or click on this little outside door. This will also naturally happen if you just leave your Apollo instance there. Okay, so don't be surprised. You should be able to see my window right now. So the username will be over here and the password here. So for the example, user.1@example.com. And then the password is just user1 without the dot. This is how you create a new annotation. You can either click, give a right click to one of those elements that are located in the evidence tracks, here highlighted with a ginormous yellow arrow that shows you how you can pull out that right click menu and show you how to start a new annotation. So you can just choose any appropriate evidence from that list of tracks. You can also drag and drop an element from the tracks area into the user created annotations area. So say if you are looking at the Apollo window and you have a gene of interest, so in this case, we're going to look at that GB51342. See that if you double click on that gene of interest and drag it up, you're creating a ghost on top of the user created annotations area. That big blue arrow is showing you the ghost. After you drag it over the area, hold it there for a second and drop it. Just drop the mouse. That action is going to create a new gene model as well. There it is. Drag it. Drop it, gene model. Okay, you can do that, you can conduct that as well. That's the most basic thing. Here's some information, very important information about the DNA sequence. Do let me know, I cannot see very well the chat, so please holler if you need my attention, okay? So this is how you reveal the sequence track. So this is the underlying DNA sequence in each one of your references. This is what the genome assembly looks like. You can right-click on the annotation that you're working with and reveal the right-click menu to zoom to base level, choose that function, or you can just keep using those magnifying glasses to zoom all the way into the DNA base pair level. You can also use the menu under the View top menu item to color the coding sequence by frame. And so that will reveal both a color over the exon of the coding sequences, as well as it will color all of the frames of the DNA track. You can do, as you see, the positive strand, so the forward strand, sorry, it's going to be revealed above the sequence, and then the negative strand is going to be revealed below the sequence. So you have both senses in there. You can actually toggle this reference DNA by using the right click. So on the upper right of this window, I have a little representation where I've highlighted one of the nucleotides in the reference sequence, and I have revealed a right click menu that says toggle the reverse turn, and the big yellow arrow shows us in the middle that the reverse strand has disappeared. As well, from the view top level menu, you can uncheck the show minus strands to remove annotations. So this actually toggles on and off gene models in either direction. You can also use the zoom in and out where the keyboard and shift arrow helps you to move up and down. Okay, so those are basic functionality that you can be doing along as you wish. So there are more simple cases that you'll come across when you're modifying your gene models. I define this simple case as a predicted gene that is correct or is nearly correct, and it's supported by good experimental evidence completely or mostly agreeing with the prediction, and the evidence that extends beyond the predicted models is assumed to be non-coding sequence. And if that's the case, there are a few very simple modifications that we can do to this particular gene model. There's an entire battery of functionality that you can conduct. So this is what that right-click menu looks like. At the very top, it shows you things on what to do with the sequence, either by revealing it, downloading it, or exporting it into GFF3. The second section shows you what to do with a particular gene model or combination of gene models. You can delete it, duplicate it. You can also create entrants, and it'll look for the canonical splice sites around the area where you're doing this. The middle section allows you to set the beginning and end of the open reading frame, as well as annotate selenium-cysteine proteins. I'll tell you about that in a second. Then you have the ability to use data from the evidence tracks to modify the boundaries of the exons in the gene model that you're annotating. You can also ask Apollo to automatically find the correct splice site by requesting that an appropriate splice donor or an appropriate splice acceptor is identified upstream or downstream of the region where you are. So how do you add an exon to an existing gene model? As I'm showing you with this representation, the bottom arrow on the left part of this image shows you that I've selected one exon and then I've dragged it up and I've dragged that ghost into the user-created annotation area. If I then move that ghost towards the gene model that's already there, the gene model is going to be highlighted by a green bar telling me, okay, it is right and it's ready to receive that additional exon. So that's one way to add an exon to your gene model. A confirmation box is going to tell you whether or not you're using an exon that is transcribed in the same sense. As an example, the arrow here is showing us that we're using an RNA-seq read in order to put an additional exon in an already existing gene model in that particular region, right? Oh, sorry. Just wanted to show that that's what that arrow did. So we're just dragging up and dropping it in the middle of the exon. It doesn't have to be at the beginning of the gene model. It doesn't have the beginning or the end. You can add untranslated regions by either stretching the boundary of the gene model that you're looking at. So how do you do this? You right click on at the edge of the exon. You do this by getting to the very front of that sequence using the zoom to base level. And then you place the cursor over the edge of the exon and it becomes this black arrow. As you can see, there are three images or three screenshots. The first one on the upper left shows what the gene model looked before I conducted the edit. Then the second one shows you what happens after I move the boundary to the left, denoted by that black arrow in the middle exon. You can also do this following the procedures that I said for adding an exon, or you can use the set as 5' or 3' end. So here's an example of doing that. In order to modify an exon, you can match the data from the predicted gene with the data in the evidence tracks. So the little yellow, thinner yellow arrows are showing in the top an offending exon that tell us this is not a canonical splice site. And the smaller yellow arrow at the bottom is showing us a number of RNA-seq reads showing where the potential boundary for this exon should be. So if I select with a shift both the read from the evidence track and the gene model in the annotation user-created annotations area, then I will be able to pull the right-click menu and tell Apollo, okay, modify the boundary in my annotation by setting the 3' end as the one depicted on the reads. I know here is the 3' end because this is a reverse transcribed gene. Okay, more on this will be, I know this is a little overwhelming at the beginning, but you'll be able to see this more easily once you're doing it on your own. And then the big fat orange arrow on the top shows the results of conducting that edit. Remember that in some cases, the data may not agree with the annotation. And in some cases, some data support the annotation and some support alternatively spliced transcripts. So annotate as many of those as you need. And then once you're done conducting every single edit, make sure that you check for the exon integrity. Make sure that you have exons from different tracks are sharing the same start and end coordinates. And I'll show you more about this edge matching function. As I was saying before, Apollo has a brain, which is the editing logic, and this is always selecting the longest open reading frame and coding sequence. So after each edit, Apollo will recalculate this open reading frame unless you pull the right-click menu and say, set the beginning of translation here, don't move it. Then so that I'm showing in the right part that if you double-click on this particular gene model, you'll be able to select the entire model. And if you do that, you notice that there are these red lines that match gene models that are in the user-created annotations area. Now I'm looking at the image in the middle. So the element in the user-created annotation area is on the top, and the element that matches it is part of the evidence tracks area, here depicted in purple. If those red lines are lit up, that means there is that the boundaries are matching. Okay, so it allows you to confirm whether the evidence is in agreement with the hypotheses, in this case, your gene. An orange circle with a white exclamation point in the middle denotes that there are non-canonical splice sites. This means that you now have an offending exon that does not have the GTAG splice that we had reviewed before. So make sure that you zoom all the way in order to review these and change to the correct donor or acceptor depending on where you're looking at it. So to demonstrate this in an example, I'm showing you here that you can adjust the boundaries as supported by the experimental data by using a similar technique than what we did for the 5' end. What you do is you go to the gene model that you're trying to modify, right click, and then ask Apollo to seek the next available splice donor or acceptor as it corresponds, upstream or downstream. My rule of thumb is always be conservative. So always go for smaller in the absence of all evidence data in support of these annotations. Okay, we can talk more about this, but basically you want to make sure that you're as conservative as possible in keeping with the evidence that is supporting your gene models. After you conduct all of these annotations, you want to make sure that you have start and stop sites that have been recovered. You can set the start if you wish to do that using the right-click menu as well. In very rare cases, you have a start codon that is not ATG. When we're talking about Munoz-Torres, that's a rare thing. So make sure that you annotate and that you write information about it. All of this information, of course, is in the slide, so you can come back to this afterwards. A little bit more complex annotations would be, for example, when you have to bring two gene models together because you know that the evidence is now showing you these two predictions are actually part of the same gene model. So you just click on one of the exons, the ending exons, terminal exons that are closest to each other from both gene models. You hold down the shift key and then you right click on one of them to reveal the right click menu to merge, or you can delete something, or you can split them apart. So you can bring these two together by using the merge function, or you can split a gene prediction because you've gotten the sequence out of your user created annotation and then realize that there are two very different motifs that are part of your annotation that shouldn't be. And in that case, you need to put them apart. So if you have a domain in one side that's a hox and the other side you have some capsays or something like that, they probably don't belong together, so you need to split them apart. The same thing, right click menu, split. You can also annotate frame shifts and you can correct single base errors. So as a note, I always try to remind everybody that what you're looking at is actually a frozen picture of the genome, and that you won't actually be modifying the assembly. Rather, you will be annotating the assembly by telling Apollo, okay, when you come to this particular nucleotide, read an insertion or read a deletion or read a substitution. And the way you do this is you right click on the actual DNA sequence, and then you select whichever function you need to conduct. If you have to add a substitution, you will write the nucleotides that will substitute, always counting to the right of the screen, no matter whether you're substituting of the forward or reverse strands. If you need to delete sequences, you'll enter a number of nucleotides that need to be deleted because you have supporting evidence. And if you have an insertion, then you'll add it by writing it on the forward or reverse strand. Just remember, you're not actually modifying the underlying sequence. You're simply annotating so that the editing logic can read those additional nucleotides, for example, and modify the coding sequence accordingly, or that it can read that some of those nucleotides have disappeared. This is what it looks like after you, for example, inserted a G in the middle of this sequence. You can also correct selenocysteine-containing proteins. You have supporting evidence telling you that that particular stop codon should be actually skipped through, and in order to read through, you just put your arrow in that particular codon, right-click to it, and tell Apollo, set a read-through stop codon in order to skip through this sequence. And as a result, you'll have a darker orange highlight that you can see on the bottom left of this image. And when you retrieve the sequence, you'll see that Apollo has now inserted the uracil in this particular region. So now you have selenocysteine-containing protein. The GFF3 will reveal this, And I'll go into more details with your admins regarding how to round trip when you export things with cysteine containing proteins. Everything that I just said over the past 6 or 7 minutes, I think, is all listed in this very, very busy slide. So don't worry about it. I'm not going to go over it. It's just that this is the summary of everything I just went through. Then let's talk about what kind of data we're going to attach. This is the last part now. Once we're done correcting the structures, what can we do in order to finalize all of this work? So we start by going to the information editor in order to add this metadata. In the information editor, you can use, include information about cross-referencing to other databases. You can information about attributes, publications, but just keep something in mind that you will have to look as a rule for your team. If the gene that you're annotating does not have multiple isoforms, so all of the metadata in the information editor is going to go on the gene side, on the left-hand side. If you're annotating more than one alternatively spliced form, then you'll have to populate the information on the mRNA or transcript side. This will become clear when we look at the information editor. For the purpose of this training, I've actually added all of the information as though I were annotating an isoform. So this is the information editor. At the top, you have information regarding whether you're annotating information for the genes, for that entire locus, or for one particular isoform, here marked by the word mRNA on the right. You can add information about publicly or privately available databases that contain the same gene. For example, I know that for this particular gene that I was annotating called SPEL1, I have an NCBI entry and I have a BBase entry for this particular species for that same gene. So you can, the way you do this is by clicking on this add button. A window like this that says enter new this or that will be revealed, and then you type the database in the accession. This is the less common information. It's a nice to have. You can also add information about literature that you've been perusing in order to define the structure and the function of this particular gene model. So you can do that by, again, clicking on the add button in the PubMed IDs, entering the PubMed ID, and in this case, Apollo is going to give you a warning and say, are you sure that this is the publication title that you wish to look at? Oh, you can't hear me?
Yes, we can.
Oh, okay. The Rosas Laboratory said that they cannot hear me.
Yes, I'm hearing.
Okay, maybe they're having some issues. You guys, you can call on the phone. Oh, how do we know if they cannot hear me? Okay, sorry about the interruption. I'm hoping that they can hear us soon. The rest of the people can hear me, so I'm going to go on. And since this is being recorded, we can facilitate that information to them later on. I'm sorry that they're having difficulties. So you can enter the information about PubMed, and they can hear me now. You can also enter information about gene ontology identifiers. So you can use those terms. We have the ability to connect to the Golar database, which is the gene ontology database. And if you type either the name of the gene term of the gene ontology term or the identifier for the gene ontology term, you'll have a little fill in the autofill that will tell you which one is the gene ontology term that you're looking for. As well, you can enter information at the very bottom, comments regarding what you did to this particular gene model. In this case, I said something like, oh, I extended the 3' UTR using forager RNA-seq reads. For your particular case, you will also have information to enter into this status radio buttons. You can say that a gene model has been approved, deleted, or it needs reviewed. And then you'll have a few other rules that are available from the BIPAA website. How do you go through all of these edits? You actually have the ability to keep track of each of the edits that you've conducted by revealing the history. You have an undo and redo button, yay, just like any other Microsoft type of editing or any other software, much of it. And to show the history, you just right-click on your user-created annotation and then click on Show History to reveal all of the annotation edits that you've conducted. All of this is stored in a centralized database. These are some of the examples that in the upper panel, my blue highlight is on the function of merging exons. And if you see that little cartoon of the gene that has been drawn, this is what that gene looked like when that particular function was conducted. Later on in the bottom panel, my blue highlight is now on the function of set the read-through stop codon. And by doing that, I've extended the coding sequence. If you look at the cartoon, you can see that now the gene model has been extended. You see that little blue line darker in the middle of the exon denoting the read-through, and then the rest of the coding sequence now. And the difference between those thin blue blocks that were untranslated region in the upper model, and now purple, green, and terrible pink in the bottom to denote the coding sequence. Okay, so now that you've gone through the whole travel of conducting your annotation, I put together my personal checklist that I go through in order to realize whether or not I've looked into all of the things that I needed to look at. Just remember, after running through the checklist, that you need to validate your annotation. Even if you've made no changes to it, just comment on it and add information that can help the community. This is a community effort, and your information, your expertise will actually be constructive to the project. So this is my checklist. Check that you have a start. Check that you have a stop. Make sure that all splice sites are canonical, and if they're not, note that they're not supposed to be canonical. Check that you have annotated all untranslated regions as the data allow. Check that you've accounted for gaps, that you have been able to merge or split gene predictions. Note that as well. And then also add important project information as comments, cross-references, or any other additional information such as gene ontology assignments. We're at the hour mark now, and I am going to run through a very, very simple example that will show you what I have in mind while I'm doing annotations. You can actually follow along. In the email that I sent you, there was a sequence file that you can use to follow along with me. These are in these you would do in the instances that I share with you. So this is going to be the honeybee genome. It's a training instance that we have with lots of data. This is a very well-loved genome, and we have a lot of information about protein coding genes that are supported with biological evidence. There's NCBI gnomon pipelines and several RefSeq-based predictions, as well as other ones that are built using ab initio techniques. In the upper left part of this slide, I am showing that we have a official gene set version 3.2, and this will be the starting point for all of my annotations in their particular example. There are several other lines of evidence. For example, there are some forager RNA-seq reads as well as nurse RNA-seq reads, which will become useful in this example as well. Those are listed under transcript sequence alignment and some other information for you to peruse. So for my particular example, I chose rhabdinase. It's an enzyme, and it cleaves fatty acids, produces this sphingosine, and turns this into a phosphorylated sphingosine kinase. So the ceramide, the SPH1, and the S1P are bioactive lipids, and are used in cell proliferation, and differentiation, apoptosis, adhesion, and migration. And this is just some background information that I needed to know in order to start working with this gene. You may be far more well-versed on this than I am. Is simply an example. And so I know in my hypothetical case that it has been brought to our attention that the honeybee ortholog for this particular gene may be fragmented into 2 or more genes. And so we're going to figure out if that's true, and if so, we're going to fix it. When you use an Apollo instance, like the ones that we have set up for our training purposes, the way you go about locating a gene or region of interest is by interrogating the genome using BLAT. We use BLAT instead of BLAST. In doing so, we're sacrificing a little bit of sensitivity for speed. So this means basically that in order to use BLAT, you'll have to use sequences that are longer than 40 residues in length, as well as sequences that are more closely related. But I'm going to run this with the example. You can do the exact same thing by going to that text file that I send you, and you can use and copy the sequence, but do not copy the definition line. So this is something to remember when you're looking for sequences using our instance, our built-in BLAD. You would have to enter the sequence in the search sequence box without a definition line, and always click on that search all genomic sequences box. After a few minutes, probably be done with the example by the time you see this table, depending on how the servers are working. But after a few minutes, you'll get a table that looks like this. And in that table, you'll have the results. So on the center of the image, you have a traditional sequence similarity results table that has the HSPs listed with a start coordinate and an end coordinate, as well as the most important value here, the significance score. So you're looking for things that are 10 to the minus 5 or smaller in order to tell you that the sequence similarity is most likely happening because of biological meaningful similarities, not just by chance. Apollo will highlight, if you click on one of these results, Apollo will highlight in this hideous yellow the region that you're looking at. And you can actually navigate to that region. Once you've navigated to the region, if you close the results, then you're done looking at results. You would have to rerun the search if you're done and satisfied with it. Now, because the BIPAA team has done such an amazing job, they've actually integrated a BLAST interface into Apollo. So you can go to the resources that Anthony was describing before. For example, if you go to Phylloxera, you have this BLAST tab, and you can enter your sequence over here. In that case, you will enter the definition line. I've done this with an RNA polymerase II sequence that I had from one of the other bees, just to try to retrieve some data. And then the results that you're going to get are going to be as depicted on the right. Your BLAST job is going to run. You'll get a list of results. You'll get e-values that will tell you how meaningful these similarities are. And then you can click on some of those to see, reveal the information that Anthony was describing before. As well, you can use that information, those gene models, for example, to come back to your Apollo instance and enter that particular name, the candidate gene, on the search box, and Apollo will navigate you to that region. So once you find the region, you create a new annotation. Remember what I said? You pick it up either by double-clicking the entire feature, or by clicking on the intern to select the entire region, and then you drag, creating that ghost in the user-created annotations area, and you drop it to create a new annotation in that region. Okay, so this is what it looks like. Just drag and drop a new annotation. If you're following along, that's what you should be able to see. If you don't see it, refresh the window. You have 30 other friends doing the same. If there are people around you that are doing the same and you can see an additional gene model in that region, just right-click to the user-created annotations area and modify the mRNA portion where it says name, and just change it to Moni's favorite gene. And then that's what you're going to see on the user-created annotation area. Here comes the train again. If we're looking at the transcriptomic data, we've now zoomed out and we've revealed sequences from the RNA-seq reads of the forager bees. And we can now see, and this is a little small, so come closer to the screen and look at that gene model that we just dragged up on the right, right part of the sequence of the screen, sorry. All of these reads, the darker images that are matching the exons, are evidence of transcription, right? And then you see these long lines. You see those? These very long lines, the lighter ones, that are connecting portions of the same read, evidence of transcription. But these are separated like 20,000 base pairs, right? So this is showing us that there's actually evidence of a large intron and additional exons that may be located way downstream that none of the prediction software was able to identify. Now you're going to say, but Moni, there's all of these other darker reads here, evidence of transcription. And that is true. Likely there's some alternative splicing that we're just going to look the other way and not take into consideration now. For the sake of the argument, we'll annotate only this one isoform. So if I zoom into this region that I have so brilliantly highlighted with terrible dotted yellow lines, then I can see that we have some of that RNA-seq reads that are supporting that exon on the right in the bottom figure. and then extensive reads that say there is no evidence of transcription in this region, or there's evidence of alternatively forms of transcription. So if I go to the left all the way, so if you look into this picture and come all the way to the left, there's a little gene model that starts at the end of that long gap. So let's zoom into that. This is now that other gene model. It's called GB40336. So we're going to do the same. We'll double click and pick it up, drag it up into the user created annotations area. And as a result, we can see all the way to the right in the top image and all the way to the left in the top image, 2 gene models. So you click on both of the end exons here to bring them together. That's how you merge 2 gene models that have been predicted that should be brought together. There's the instructions on how to do that. As a result, you're now going to have non-canonical splice sites, because the prediction for both of those genes was that they were separated and that the stop codon for the one on the right, the one that was original 40335, ended about 1,400,000, and then the other one didn't start until about 300,000, right? So they're quite far apart. So in order to correct those, you'll simply zoom in. First of all, by looking into this area, we're seeing that the exon's not supported by the RNA-seq data. So the most logical thing to do in here would be to get rid of that offending exon by right-clicking and deleting. See that all of these empty space says there's no supporting of transcription, so let's get rid of that exon. And then on the other end, we have an offending exon with a non-canonical splice site. In the image all the way to the right, you see the edge of the exon with an orange circle. So there is also there are all of these RNA-seq reads in support of transcription ending somewhere far after this particular one has been said, right? So this is all reverse transcription, so that's why I'm saying after that predicted exon. So I can use one of the reads from the RNA-seq reads and shift click on my annotation in order to set the 5' end of this exon to emulate what the evidence of transcription is showing us. So this offending exon is now modified, where we can do it that way, or we can actually just right click on the gene model and say, okay, set the downstream splice acceptor. So we're actually asking Apollo to automatically define that boundary. In that way, we're fixing the non-canonical splice site. This is what it looks like after that fix has happened. And what we do then is to corroborate whether or not we have rescued a coding sequence that is useful. So we'll go to get the sequence in query publicly available databases, in this case, BLAST from the National Institute of Health in the US. And I'm querying that database by using a protein sequence to query a protein database. So just BLAST-TB in this case. The way you retrieve the sequence is by selecting the entire model, right-click and get sequence. You have the ability to retrieve just the peptide, just the coding sequence, the cDNA genomic sequence, or genomic sequence with boundaries in case you're looking for some regulatory regions as well. The results of doing this versus NR, for example, shows us a putative conserved domain that recovers the ceramidase domains. So remember, that's the gene that we started with. So yay, it seems like we've actually recovered the entire domain in the little ceramidase alkaline superfamily domain as well. The list of HSPs here shows us some of the examples, and it seems that we have both good coverage and significant e-value for all of the hits. So that's useful information. Once you're done, you can use all of the information that you found to add metadata. So we'll include, remember, you must include the name of the gene, the symbol. If you wish, you will include some description. And in the case for BIPAA, if we were annotating more than one isoform, then on the right will annotate mRNA, on the left will annotate gene if you only have one splice form. You can enter information like the PFAM family domain and other data for your information editor. As well, you'll enter PubMed identifiers, gene ontology terms, and comments, in this case, the product of merging 2 gene models. There are some rules specific to the BIPAA site. I'm now officially a minute over the time that I had allotted for this, so I'll go through these quickly. Thankfully, they're all listed on your website. As Anthony was saying before, each genome hosted on BIPAA has a dedicated homepage. If you don't have an account, you'll create it from there. This is the URL for the phylloxera, but you need to have permission in order to access those resources Anthony mentioned before. A computational predicted consensus gene set has been generated, and that's using Maker. Just remember that if you're working with the gene annotations, if you recognize that something needs to be deleted and you don't delete it, it will make the official gene set. And if you recognize that something should be there but you don't put it, it won't make the official gene set. So it's up to you that the most accurate representation of the biology passes on and that your team has the best representation of that genome. There are a few other rules for this particular curation process. Remember that in some cases, the metrics that are used to generate the sets actually reduce the accuracy of a gene representation. So be cautious and use your better judgment. I am here reposting the rules about isoforms that I've already said twice, so I won't say it again. If an annotation needs to be removed from the consensus gene set, just make sure that you drag it in the user create annotations area nonetheless, and that you copy the gene identifier and enter the delete radio button. These and other rules are available on this website, How to Annotate a Genome from the BIPAA site. Okay, so you can just access all of these rules that I've listed here from there. Should you come across a gene of interest that somebody else is already annotating and you really, really want to annotate it, send them an email. That's why this is a collaborative effort. You can find the common place and you can actually hash out the best representation of that gene. As Anthony mentioned, there are annotation reports, so I won't repeat this. And there are processes in which you are continuously updating this OGS. It'll happen regularly. So you'll have that report to tell you whether things have happened correctly or not. I'll discuss this question with you guys later. I think I sent Anthony an email about overlapping CDSs. All of the information that you're looking into this training instances is also available in our Honeybee demo, available from genomarchitect.org. So you can access the same data once I bring my Amazon machines down tomorrow morning. And you also have a little demonstration video that's available from YouTube in case you are not tired of listening to my voice after an hour and a half. This is the team. I'm the project manager for the Apollo project. We work at Berkeley Bioinformatics open source projects. Nathan Dunn is the magician software engineer behind the developments of the new architecture for Apollo. Susie Lewis is our investigator, who, by the way, just sent me a text message to let her into our office. And Eric Yao is the lead developer for JBrowse. Deepak Uni is a magician at University of Missouri under Chris Elsix, without whom we would not be counting our blessings here. And thank you very much. I'll be happy to take some questions, but I know we've gone now 3 minutes over the time that I had destined for this. Thank you.
Thank you.
Yeah, thank you a lot, Monica.
Thank you.
Any questions?
Yeah, I have one quick question. Can you hear me?
Yes.
This is very basic, but when you were talking about the different annotations and where there was mRNA evidence, I'm just not familiar with all the colors. Is there like a really beginner's guide for what is mRNA and what is protein evidence, or are these not logical?
You mean the colors and the actually
Yeah.
these are just things that we chose. It doesn't need it doesn't have. I'm trying to go back to what that window looks like so that you can see them. There aren't any specific patterns. We
Okay.
try to do them. Yeah. So the three colors that are said in this particular instance so are only the ones that are defining coding sequences. So
Okay.
terrible green, terrible lilac, terrible pink are colors that we chose that you cannot change, or you could, your administrator could. The other ones are also represented differently. You could just choose that all of the aligned metazoa are colored in purple or gray, and you could choose.
So maybe I just missed why some of that greeny dark teal was grayed out and some wasn't.
Correct. So remember, so that particular I'll bring one that has a little zoom in into those regions. Where did it go? So for example here.
Yeah, exactly.
Correct. So what you're looking at here, remember, are representations of evidence of transcription. So you went and created your libraries, you got some RNA-seq reads. These are all reads of messengers of RNA, which means introns have already been spliced. So when you align those reads against your genome, what you're going to get are these darker regions showing you the actual reads, the actual transcription, but because they came from already spliced mRNAs, then the rest of the particular read is going to be located somewhere downstream.
Okay, so those gray bits are just inferred gap. I see.
Correct.
I just couldn't see where the rest of the read was. Okay, I understand. Thank you very much.
Yeah, sure. Okay, we have some chat questions. Oh, that was just a bunch of thank yous. Thank you, guys. And somebody was asking, how do you visualize so much RNA-seq data at once? And so I have a few tricks up my sleeve because I know that you're not able to see all of those. For when you get, Anthony and Fabrice are probably not going to like me for showing you this, but I will show you nonetheless. Here they are. So if you're looking into an RNA-seq track, in this case, I'm going to bring some RNA-seq reads from nurse bees. So what you do, this is for, I believe, for Quello, is there's a little JSON file that you can modify, or did you change the feature scale? And so you can do this. It's not a permanent change. It's only a change for visualization. So if you change the feature scale here to 0.1 or 0.01, for example, then let's give a headache to this particular server. Then you'll be able to zoom out and still see lots and lots of RNA-seq reads. And you'll zoom out and you'll still be able to visualize them. You can also change the maximum height for display of these sequences from 1,000 reads to 2,000. It'll just load your client, and eventually your client will just crash. I hope that answers your question. And thanks everyone for the little thank you messages. I will be available through email as well whenever you guys, if you have any question. And through the curation process, I will be also happy to assist the BIPAA team to help you with a few questions. There are some edge cases that will come about, and we'll be discussing with Denis and Fabrice and Anthony about the best way to model those edge cases. So feel free to ask us. Always copy everyone from the BIPAA team whenever sending a question so that they're aware of that communication.
Yeah.
All right, you guys were awesome. Thank you very much for listening. And this is all being recorded, so I will make sure that this is available to you guys. I may edit some of my bad jokes.
Yeah, we can add the video on the BIPAA website, I think. We'll have to discuss it by email. We'll see.
Sure. We'll figure that out. We can do it there. I also have a curation resource that we may be able to upload it to, or we can just put it on, make it available on YouTube. That's fine with me too.
Yeah. Yeah. Okay. Okay.
Brilliant. I'm going to stop this then. Thank you very much. I hope everybody has a wonderful rest of the day, and make sure to email me if you need anything.
Yeah,
great. Thanks very much.
A lot to you.
Thank you. Thank you.
Yeah.
Sure. Happy weekend.